monocytes. Okay? They are low inflammatory. They can be certainly stimulated to become inflammatory, but they're classical. They're not really inflammatory. You have low pro-inflammatory monocytes, and you even have high pro-inflammatory monocytes. A colleague of mine has shown that consistent resistance and combined resistance and aerobic training reduces the proportion of these high pro-inflammatory monocytes in the body. So we speculated that maybe that's why we were seeing more or less cytokines in the first study. It was actually less. So why are we seeing them here? We did not measure the different monocytes in that first study. So we're doing that now. But I want to get back to these last results. You asked if the increased TNF-alpha is good or bad. Okay, well, we also measured IL-10 in those same cultures. And of course, it is the big anti-inflammatory cytokine I mentioned. Here's what we found there. Again, we measured before and after exercise, 2 hours after exercise, and 24 hours after exercise. Let me start down here. There was a training by time interaction that just simply showed us there's a diurnal reduction here as well in IL-10. So there are certainly diurnal factors. You have to have a control group to tease apart any type of effect of an intervention you're studying. There was also a group by training after exercise where the exercise mean was greater than the control mean. So it shows the mean of the C's. All of these are the gray C bars are after training in the exercise group. Okay? Notice that this is before and after training and control. They actually experienced a reduction, an overall reduction in IL-10 release. So now what's going on? We had to consider all the data together. Overall, in the circulating blood, we saw reductions in CRP, TNF-alpha, and leptin. That's good. In the stimulated cultures, we saw an improvement in TNF-alpha, which is we originally thought was pro-inflammatory. And then we also saw, though, an improvement in IL-10 in the exercise group, but a reduction in IL-10 in the control group. So we've interpreted that to indicate that exercise actually improved the immune response to the mitogen in obviously the exercise group, whereas something else was occurring, maybe a shift due to seasonal variation from to a more pro-inflammatory state in the control group, because they're producing less of the anti-inflammatory IL-10 and more of the other inflammatory cytokines. And we found out this is supported in the literature because morbidly obese individuals actually experience a reduction in immune function. Okay? There's a, I don't have a slide that I can show you really quickly. Maybe. This is called the J phenomenon. Over here, risk of upper respiratory tract infection. Everybody knows what upper respiratory tract. And this is training volume. This is a healthy but sedentary person. So a healthy but sedentary person has this much risk for a URTI. As they begin an exercise training program, exercise reduces their risk of getting sick by improving numerous different immunological responses. However, if training gets really high, actually, as in collegiate folks doing, or Olympic folks, it actually increases their risk of URTI. But obesity has the effect of simply, well, doing this. At least morbid obesity. It was shown that those who are morbidly obese can improve immune function or reduce risk of disease by losing the weight and therefore improving their inflammatory cytokine production. So there are a lot of factors that influence. You can't just say, well, IL-6 went up. In the blood, maybe that's okay, but in stimulated cultures that measure function rather than just a static, not necessarily static, but rather than just a concentration in blood, these more functional assays help to give us a better idea of what's happening. And I think that our data, even though they weren't morbidly obese, I think we can say that it is probable that exercise improved their immune function, overall resulting in a reduced state of inflammation. So here's the summary for this study, at least so far. The reduced circulating markers we just mentioned, improved immune response to an endotoxin challenge, increased LPS as well as the IL-10, and possibly a more pro-inflammatory state in con due to that decrease in IL-10 concomitantly with an increase in TNF-alpha. So I hope that makes sense. So we wondered, why is this happening? What is the mechanism by which exercise is reducing inflammation? I have to give kudos to a colleague's graduate student brought up this system called the melanocortin system. Everybody knows what melanocytes are. They help give you a tan, right? That's part of the melanocortin system. It does 1,000 other things, though, as well. From controlling eating behavior, fat distribution among the body, sexual behavior, and inflammation. Okay. All of these, this is pro-opiomelanocortin hormone produced in the brain. It is converted to various other melanocortin molecules or hormones. One is ACTH, you may be familiar, part of the hypothalamic pituitary adrenal axis, stimulates what production in the adrenal glands? Cortisol, yes. Okay, that's a little metabolism there. But it also will bind to these melanocortin receptors. So what happens? Well, these different hormones circulating will bind to their receptor on an immune cell, like in a macrophage or monocyte, other inflammatory cells, and they block the whole process of chemokines stimulating the inflammatory response. So this is just this little chart, just to know that they help to block the inflammatory response. So they're a type of regulator. There are 5 different receptors, depending on what the tissue is. And the ones we were most interested in were the number 1 and number 3 receptors. They're found in, they're most numerous in monocytes and other leukocytes. Again, they respond to their activations, all of these, by reducing pro-inflammatory cytokine production. And key, this is really key, they increase anti-inflammatory cytokines, specifically IL-10. So we said, well, our IL-10 went way up, along with TNF-alpha in the exercise group. We got it from melanocortin. Well, we don't know, but we didn't know. But that's what we hypothesized. So, kind of post hoc, we took some of the stored whole blood and measured gene expression of one of those receptors, the MC3R gene. Okay? So, this is in whole blood. We used real-time PCR, polymerase chain reaction, and we found that there was no significant change in the control group, but the exercise group experienced a 16-fold increase in MC3R expression in whole blood. So, that's it. No one had ever, at that time, well, still not actually, examined MC receptor expression to exercise, the response to exercise. So we decided to follow up very quickly. This paper's in print at Journal of Aging Research. To our current study, to further investigate if fitness or exercise really does influence it. We decided to go first, before another training study, just take a cross-sectional study and examine physical fitness and obesity on the melanocortin system, regulation of inflammation in humans. We hypothesized that both of these anti-inflammatory receptors would be upregulated in fit groups, okay, versus sedentary groups. We'll talk about that in a second. We hypothesized that obviously inflammatory reactivity would be elevated in the sedentary versus the fit, and the obese versus the lean. So what we did, we ended up with a 2x2 design. We'll talk about those in a second. Let me just show you the design. Real simple. 4 groups, it was a 2x2 design, 20 subjects per group, 10 male, 10 female. Here the age, no oral contraceptives or other hormones, no dyslipidemia meds, and apparently healthy, no diabetics. So we had a lean, sedentary group. We're working real hard on that group, actually. Physically fit, lean and fit, or normal weight. You could call either of these normal weight. And then we had obese, sedentary, and obese fit. This way we could look at the interaction between adiposity, fat, versus fitness. So what we're doing, we're measuring inflammation in several different areas. First of all, in circulation, in the blood. These are some of the markers we're measuring. Just to get a blood sample, measure these markers. We are also measuring these inflammatory cytokines from the culture method I described previously, with one little zing in it, I'll show you that. And then we are also getting gene expression from whole blood, okay, using, again, real-time PCR, thanks to Dr. Cheek and Gary Vaughn. The real-time is in Gary Vaughn's lab. And we'll do this on fresh whole blood, as well as these stimulated cultures, the blood cells that are left over there. So we're going to look at what's going on with these receptors, not only in fresh whole blood, but also in the blood that is stimulated. And then finally, a fourth big, nice, awesome type of analysis is we'll be measuring the cell surface protein expression. So gene expression tells you what's going on in the nucleus. Is that gene active? However, it doesn't necessarily translate to whether or not you're producing the proteins onto the surface of the cell. So using a process called flow cytometry, thanks to Mike Chumley over in biology, we're using the flow cytometer, we can actually measure these receptors on the cell surface. So, I don't know. The more you got, you can, actually the more you have more color development. I'll show you just briefly what that means. So we're doing that also on fresh whole blood and on the blood cells that come out of those cultures. So we've got a nice overall complement of analyses to try to really investigate this. This is the same slide you saw before. Here's the one zing we added. So not only are we adding LPS to the blood samples here, we are also adding or not the alpha MSH hormone that would stimulate those receptors on the cell, the MC1 and the MC3 receptors. Such that, as an example, if we have a cell or a well on our culture plate with a fit person's blood and you add LPS, well, it's going to produce a lot of cytokines. If you take that same fit person's blood, you add alpha MSH, we think the cytokine production will be very blunted, more so than the same well in an unfit or obese person. Meaning the sedentary or obese individuals will not have as great of an anti-inflammatory response as the fit lean. So we have a whole matrix set up to measure this. The rest is the same. And I want to point out one more thing so I can show you some, actually we're almost done, show you some actual data from this particular study. We can't do the gene analysis yet or the cytokine analyses because we keep all those blood samples frozen until the whole study is done so we can run them all together. But the flow cytometry, where we measure the cell surface markers, it has to be done on fresh blood. So I have some of those data to show you. We notice in the literature that most of this has been looked at in mice, if at all, not in humans. And no one has ever investigated the difference in the melanocortin receptors on these different monocyte populations I mentioned a minute ago. So again, we have classical monocytes. How do we know it's a classical monocyte? You can't tell just by looking at it. You use flow cytometry. And if it has this cell surface marker known as CD14, that means it's a monocyte, period. So we can identify it as a monocyte if it has CD14, and we can measure all those CD14s on the cell surface. If it does not have CD16,